Welcome to the Genetics series. This video focuses on Punnett squares. At the end of this video, students should be able to perform monohybrid and dihybrid crosses and identify the gametes formed from the parental generations. The monohybrid cross looks at one trait with two possible alleles. They form either the homozygous dominant genotype, the homozygous recessive genotype, or the heterozygous genotype, which is also expressed as the dominant phenotype. Let's look at an example. If we have a parental generation of true breeding pea pods, this means they are homozygous for their allele. If we arbitrarily assign the letter G to pea pod color and give the green pods big G as the genotype, we would use small g for the yellow genotype. When we do our Punnett square, we have 100% of the offspring in the F1 generation with big G genotype. When we observe the phenotypes, they are all green. This tells us that the green phenotype is dominant and the yellow phenotype is recessive. Let's try another problem. This time we will look at seed color. If we cross two heterozygous yellow pea plant seeds, the genotype for each would be big Y, little y. Although I would try not to use Y since it looks the same in upper and lower case, especially with handwriting. When we cross the heterozygotes, we would expect a 3:1 ratio of dominant to recessive in the phenotype. This means in our Punnett square that 3 out of the 4 would be yellow, and 1 out of the 4 would be green. Statistically, what this means is that if you have a 3 out of 4 chance of creating a yellow-seeded pea plant when mating two heterozygotes, that's 75%. You would have a 1 out of 4 chance of creating a green-seeded pea plant, which is 25%. Scientists can also look at two traits at one time in what is called a dihybrid or two-factor cross. In order to determine the gametic combination of alleles for dihybrid crosses, we use the FOIL method. Just like in math, FOIL is first, outside, inside, last. So you choose one parental genotype and FOIL it to determine the possible allele combinations or genotypes for the gametes. In the instance of big A, big B, little b, we could have big A, big B, or big A, little b, or little a, big B, or little a, little b. In the case of big P, big G, we could have big P, big G, little p, big G, or little p, little g. These are the allele combinations that each parent could possibly contribute to make up the sperm or egg cell. We write each one set of the parental allele letter combinations at the top of each column, or alongside each row, similar to the way we do in the monohybrid cross. Let's look at the parental pea plants with regard to the two traits seed color and seed texture. The seeds have either yellow or green coloring, and we will choose big G to represent the trait color because unlike Y, it looks different in the upper and lower case forms. The texture of the peas are either round or wrinkled, and we will choose big R as the shape trait over W, since again, big R looks different in the upper and lower case forms. For color, we will attribute big G to green and little g to yellow. For texture, we will attribute big R to round and little r to wrinkled. I always make myself a key to remember which alleles represent which phenotypes. In the parental generation, or P1, we will cross a true breeding round yellow seed plant with a true breeding wrinkled green seed plant. Because we know both of the strains were true breeding, we know one parent was homozygous dominant for color, and the other parent was homozygous recessive for color. We also know that one parent was homozygous dominant for seed shape, and the other parent was homozygous recessive for seed shape. We can assign parent 1 big R for round, and so parent 2 will be assigned little r for wrinkled. We can assign parent 2 big G for green, and parent 1 little g for yellow. This means we would have little g, big R times big G, little r. The first parent with the yellow round phenotype and the little g, big R genotype would only be able to give a little g and a big R to each offspring, since when we FOIL it, they all result in small g, big R. The parent with the green wrinkled phenotype and the big G, small r genotype would only be able to give a big G and a small r to its offspring, because when we FOIL it, the genotypes available will all be big G and small r. As we cross the parents using our Punnett square, we know that all the offspring will be doubly heterozygous, or both traits would be heterozygote. The genotype for the F1Ps would be big G, big R, little r. The cross between a round yellow parent and a green wrinkled parent produces 100% yellow and round phenotype in the F1 generation offspring. This shows us that the dominant phenotype in seed color is yellow, and the dominant phenotype in seed shape is round. I like to note by my key which traits are assigned as dominant and which are recessive. Traditionally, we would assign the capital letters to the dominant traits and the lowercase letters to the recessive traits. However, in this case, we will just continue to use the annotations that we have already begun using. In order to determine the possible gametes for a double heterozygous genotype, we must use the FOIL method. Big G, big R, little r can create 4 possible combinations. They are big G, big R, big G, little g, big R, or little g, little r. If we SIB mate the F1 plants with phenotype round and yellow, depicted by the double heterozygous genotype, big G, big R, little r, we will yield another predictable Mendelian pattern. The F2 generation will contain 9 out of 16 double dominant phenotypes, followed by 3 out of 16 with 1 dominant and 1 recessive phenotype, followed by another 3 out of 16 with the opposite trait as dominant and the opposite trait as recessive, following up with 1 out of 16 being the double recessive phenotype. In our case, this creates 9 out of 16 yellow round peas, 3 out of 16 yellow wrinkled peas, 3 out of 16 green round peas, and 1 out of 16 green wrinkled peas. The F2 generation in a dihybrid cross has a ratio for Mendelian patterns. It is 9 to 3 to 3 to 1. So how did we get to 9 to 3 to 3 to 1? First, we set up a 4x4 grid. Next, we assign each possible gamete and allele combination to the parent rows, mom for the rows, dad for the columns, and then we fill in the table. Each row has 4 squares, so the parental genotype for that row fills in all 4 squares. Each column has 4 squares, and the parental genotype for that column fills in all 4 squares. Each F1 possible offspring combination is now created. To calculate the ratio of allele combinations of genotypes, we would combine any identical genotypes for each possible combination. In this case, there is only one big G, big R, big R. There are 2 big G, small r, big R, 2 big G, big R, 4 big G, big R, little r, 1 big G, little r, 2 big G, little r, 2 little g, big R, 1 little g, big R, and 1 little g, little r. The pattern I try to remember is 1 to 2 to 1, 2 to 4 to 2, 1 to 2 to 1, which totals 16 squares. Sometimes when I have to write it, I write a 4 in the middle, and then I just remember from there, the pattern is the same. It goes 4 in the middle, and then on each side, 2, 1, 2, 1. And that way I can remember all of the different combinations that I'm looking for with regard to genotype. I generally put a check mark in the corner once I've used a genotype to help keep track of what I am looking at. For phenotypes, I like to identify them in a way to make it easier to group them together. I often use different colors as long as they're available, or I'll use various shapes in the corners of the squares, and I just make a key for myself by the phenotype so I know what I'm looking at. In this case, our options are less diverse than the genotype, so we only have 4 possibilities. We'd have green and round, yellow and round, green and wrinkled, or yellow and wrinkled. So I made green round a green circle, yellow round a yellow circle, I guess they're more oval, green and wrinkled a green rectangle, and yellow and wrinkled a yellow rectangle. In this case, we want to say yellow is dominant and round is dominant, so I always label those first. Anything with 1 or more G and 1 or more R will be dominant, doubly dominant. And we know from the ratio we expect 9 of those. So I place the green round circles around anything with 1 big G and 1 big R. It does not matter if it is homozygous, dominant, or heterozygote because they all express the same phenotype. Next, I choose one of the combinations that will result in 1 big G and 2 small r's, or 1 big R and 2 small g's. In this case, we'll use yellow and round. So I go through and find little g with 1 big R or more. And I put the yellow circle or oval around them. Then I come to the flip of that, where I'd be looking for the green and wrinkled. In this case, I need 1 big G or more with 2 small r's, and I put the green rectangle around those. Finally, the double recessive, little g, little r, little g, little r, we know will only be 1, and we put a yellow rectangle around that one. The ratio shows, as predicted, the 9 to 3 to 3 to 1, with 9 being double dominant, so all green and round, the 3 being 1 dominant and 1 recessive, so yellow and round, then the opposite dominant and recessive, so green and wrinkled, followed by 1 doubly recessive, yellow and wrinkled. To determine if the 2 genes being studied assort independently, as simple Mendelian genetics indicates, we would want to see a resulting 9 to 3 to 3 to 1 ratio in the F2 generation. If we have a parental cross between a yellow round and a green wrinkled, and we see the F1 generation as 100% yellow and round, we know that both yellow and round are dominant. In the F2 generation, if we find 315 yellow seeds, 101 green wrinkled seeds, 108 green round seeds, and 32 green wrinkled seeds, in order to determine if these genes assort independently, we first total all the seeds. We then multiply the total times 9 out of 16 to determine the expected number of dominant seeds. We multiply the total by 3 out of 16 to determine the expected total for both of the 1 dominant, opposite recessive traits. Finally, we multiply the total number by 1 out of 16 to determine which number of seeds are expected to be both recessive traits. These are the expectations that we compare to the actual results from the table above. The total in this case is 556. Multiplying that by 9 out of 16 gives us 312.75. Since we cannot have 0.75 plants, we must round to a whole number, so we would use 313 plants as the dominant phenotype for our expected. For the recessive plant, we multiply that 556 total times the 1 out of 16, which is giving us 34.75. Again, we'll call it 35. For the remaining phenotypes, one dominant, the other recessive, and then one recessive and the other dominant, we would expect the same number. Both would be 3 out of 16. So if we multiply that 556 times 3 out of 16, we get 104 seeds. If we compare the observed and the expected phenotypes, we can see that they assort independently. 313 expected round and yellow, 315 actual, pretty darn close. 104 yellow wrinkled by 101 yellow wrinkled, again pretty close. 108 green round compared to 104 green and round, we're almost there. And then 35 green and wrinkled compared to the 32 from the table. So when we look at this, we can see they are pretty much independently assorting. They're not linked genes. However, in lab, we will undergo a special statistical analysis of these numbers that would determine with finality that yes or no, these genes are linked, and it's called a chi-square. Comparing the observed and expected phenotypes allows us to determine if, as Mendel observed, genes assort independently. This means that the genes are not linked. So if they are located on the same chromosome, they do not stay together during meiotic division. Students should be able to perform monohybrid and dihybrid crosses. Students should be able to understand how to identify the expected phenotype and genotype of the F1 and F2 offspring. Students should be able to identify parental alleles based on genetic offspring gametes. And students should be able to determine the gametic combinations from parental genotypes.